Hello, my name is Courtney Breen, and I will be doing my graduate project on the link between gut microbiota composition and childhood obesity. The article that researched this topic was called Differences in Gut Microbiota Composition Between Obese and Lean Children, a cross-sectional study. The authors are listed below. The research question of this study was, what are the differences in gut microbiota when comparing the gut composition of obese children compared to lean children? And the objective of this study was to utilize fecal samples of obese and lean children / adolescents to determine differing concentrations of gut bacterial species. This study focused on how the gut microbiota composition is different in obese children than it is in lean children. An infant's gut microbiota starts becoming influenced during pregnancy because the health of the mother directly affects what gut microbiota will be transferred to their child. If the mother is obese and has a dysbiosis of gut bacteria, then the baby will be born with similar or the same. Vaginal birth, C-sections, and breastfeeding and formula feeding affect the infant's bacteria composition in the gut. As we have discussed in class and the article discusses, the rates of child and adolescent obesity is high. A child being obese leads to major comorbidity concerns in the future like metabolic syndrome, type 2 diabetes, premature cardiovascular disease, liver disease. Unfortunately, if the child is obese at a young age, there is a strong correlation that the child will then also experience obesity in adulthood, and the rates are increased if the parents are also obese. One can hypothesize that this is due to the child eating similar diets as their parents and being influenced by their parents during their modeling phases of their life, as we learned in class. With all this, it is obvious that the prevention of obesity is crucial for good health. Usually, therapeutic strategies are encouraged, such as lifestyle changes. Lifestyle changes can include dietary changes, exercise implementations, etc. Due to the failure rates of therapeutic changes, pharmacology and bariatric surgeries are being utilized to treat obesity. But do we even understand the breadth of obesity? The research on the gene-environment interactions that lead to obesity are a very unexplored part of the environmental factors that feed into the pathogenesis of obesity. The concentrations of different strains of gut bacteria are involved in energy regulation and inflammation factors. A significant quote from the study states, "Energy intake affects gut microbiota composition, and it is unclear if the gut microbiota plays a causal role in the development of obesity in humans." This study attempts to delve deeper into the understanding of this causal role. Now, past studies have been conducted in humans and mice, and they have found different results of the concentrations of gut bacteria in obese and lean children, and due to these differing results from past studies, this paper will delve deeper into comparing gut bacteria species and fecal samples of obese and lean children and adolescents. So how is this study designed? Well, this study is a cross-sectional study, which means that it's an observational study that measures the outcomes of the sample at one specific point in time. The study used 25 overweight / obese people and 25 lean people. The age group of the subjects was 6 to 16 years old. This includes the classification age groups of children and adolescents, as we learned in class. The subjects were all similar in terms of age, gender, height, and food intake. They took fecal samples from these subjects and utilized several different tests to identify the concentrations of the gut bacteria within those samples. The tests that were performed will now be discussed. So quantitative plating was utilized. The plated wet fecal solution was placed on agar and then measured the concentrations of the bacteria species that grew. The number of colonies were then expressed relative to the total number of colonies that the researchers examined. The second test they used was MALDI-TOF-MS analysis. Once the strains were identified, mass spectrometry was applied, which then identified the different species of Bacteroides fragilis group. So MALDI-TOF-MS is when each sample, 10 of the colonies of Bacteroides fragilis were then subcultured and measured by matrix-assisted laser desorption mass spectrometry. The relationships were then identified using a biotyper software. A final significant test that was used was quantitative PCR. This is when they collect the fecal samples and then those are selectively plated. Once plated, they utilized real-time PCR to gather the exact concentrations. The PCR determined the 5 types of readable bacteria were Bacteroides, Bifidobacterium, Clostridium, Staphylococcus, and Lactobacillus. The Mann-Whitney test was then applied, which is a significant test because it is what identified the difference in concentrations of the previously mentioned bacteria strains between obese and lean children / adolescents. So, what significant findings did this Mann-Whitney test discover? When comparing the obese subjects to the lean subjects, the following was found: elevated levels of Firmicutes to Bacteroides ratio, lower proportions of B. vulgaris, and high concentrations of Lactobacillus. Another important finding to note is that there is a significant positive association of plasma HS-CRP found in obese children. Now let's discuss the important conclusions of these results. As we know, obesity can lead to comorbidities like type 2 diabetes. Why is this notable yet again? This is because children with type 2 diabetes have low proportions of B. vulgatus, and low proportions of B. vulgatus was found in obese children. Therefore, it is enough evidence to conclude the correlation of this low proportion of this strain can contribute to not only obesity, but also the unfortunate comorbidities like diabetes type 2. So it is a large contributor to these previously mentioned chronic diseases. We will discuss the following aspects later, but this leads one to understand why it is important to maintain healthy proportions of B. vulgatus. Plasma HSCRP specifically is a C-reactive protein test. These biochemical markers from a blood test measures the inflammation in your body. It can evaluate one's risk for cardiovascular diseases. Increased levels of plasma HSCRP found in obese individuals mean that those subjects have increased internal inflammation, therefore greater risk for cardiovascular diseases, or an indicator for current cardiovascular diseases. The increased CRP levels was positively correlated to increased concentrations of lactobacillus and also an increased measure of Firmicutes to bacteroidetes ratio. What does this mean? Once again, concentrations of these bacteria in obese children is positively correlated with biomarkers that indicate internal inflammation that leads to or indicates cardiovascular disease. The findings give us insight into the pathophysiological process of obesity. We will now visualize the differences in the results via the following figures. So figure 1A above demonstrates the differences between the gut microbiota species between obese and overweight children, demonstrated in gray, and lean children, demonstrated in white. Remember, the researchers utilized quantitative plating to get these results. Bacteroidetes, fragilis group, and Clostridium were not significantly different when comparing between obese and overweight children versus lean children when utilized quantitative plating. Figure 1b used multi-top MS analysis. This method revealed great proportions of B. fragilis and B. theta-thiomicron in fecal samples of obese/overweight children, demonstrated in gray, compared to lean children, demonstrated in white. B. cacae, B. ovatus, and B. vulgatus were dominated in leaner children, and obese/overweight children had lower concentrations of those. It is important to note that the main significant difference that was discovered in this method analysis was B. vulgatus species. This bacteria is dominating in lean children when compared to overweight/obese children. In Figure C, the results were from using quantitative PCR. This method demonstrates differences of gut bacteria in subjects. It is interesting to note that the obese and overweight children, as demonstrated in the gray bar, had higher levels of lactobacillus when compared to lean children, demonstrated in the white bar, when you're using qPCR. There is more of a significant difference when using qPCR between the lactobacillus levels than when you compare this to the quantitative plating, as we saw in Figure 1A. Figure 2 utilizes a box plot to demonstrate the ratio of Firmicutes to bacteroidetes between obese and overweight children and lean children. The results demonstrate how the ratio in obese and overweight children favored Firmicutes and showed lower levels of bacteroidetes. This confirms the findings from previous research that we discussed in the beginning of the presentation. So let's recap. Obese children have an increased ratio of Firmicutes to bacteroidetes, low concentrations of B. vulgatus, high concentrations of lactobacillus, and increased plasma HRCRP levels. Presence of Staphylococcus spp is found to be associated with higher energy intakes. The bacteria strains and their specific levels are associated with childhood obesity and comorbidities that go along with them. The researchers concluded that dysbiosis of gut bacteria, along with poor lifestyle behaviors like lack of exercising, lack of healthy food, lead to childhood obesity of children and adolescents. They also indicated that if one transforms their gut bacteria to be healthy concentrations of significant strains, one can prevent or treat obesity. Knowing this, the field of medicine can help dedicate resources to preventing dysbiosis in obese/overweight children so that they are able to live long and healthy lives. While I thoroughly enjoyed the research findings from this study, I also found many flaws. For subject flaws, they utilized a small sample size. They also did not thoroughly ensure that the subjects were very similar. They never mentioned race or socioeconomic status. They did not inform how many subjects were from each age group, and they stressed the importance of maternal nutrition throughout pregnancy for gut microbiome, yet did not make sure that that factor was similar across subjects. They also noted that the subjects had similar dietary intakes, yet did not explain what they meant by that. For example, how does a lean child and a morbidly obese child have close to the same dietary intakes? They included overweight, obese, and morbidly obese children in the same category, which may have been too broad. They did factor in adiposity rebound for increased BMIs of subjects from 6 to 7 years old, as we learn in class, and that means that these children could actually not be obese despite their BMIs. Finally, the researchers did not factor in the activity levels of the subject. The researchers used BMI as an indication of lean versus overweight / obese / morbidly obese. As we have discussed in class, this can be a troublesome indication due to BMI not factoring in muscle composition, race, sexual maturation, etc. I wonder what could be a better indicator to use. Finally, the researchers did not make recommendations on how one can fix or solve their dysbiosis issue. For example, could the subjects take prebiotics or probiotics? What type of foods should the subjects focus on eating in order to improve their gut microbiota composition for the better? I believe that this study should have explained what exactly could be done to treat an unhealthy gut in terms of diet and supplementation. Despite all the flaws, this study emphasized the importance of gut health in children and hopefully can lead to the medical community focusing more on treatment and prevention efforts with a gut microbiota perspective. These are the references.